Okay, so this is going to be a comparison of gene expression in eukaryotes versus prokaryotes, right? So gene expression in eukaryotes versus prokaryotes. And I think if I had you all in here, and if we, if it wasn't a pandemic, I would have you having different colored markers on these whiteboards, and I'd have you brainstorm first before I do this for you. But things being as they are, I think I'm just going to present this to you and hope that it all still goes in. So we learned about eukaryotes last time. Eukaryotes first. Eukaryotic gene expression. Remember that? Okay, so down here, and I'm going to draw this rectangle, and it's going to look like just a simple old rectangle, but we're going to add a lot of things to it. So I want you to pretend that what I'm drawing here is a strand of DNA. That's what it is. It's not a rectangle. It is a double helix in there. Okay, if you can imagine that. So eukaryotic gene expression is kind of like the diagram I showed you last time. So if you have a gene, let's say that we have a gene here, and here we have a terminator section that tells the RNA polymerase to stop, and then here, ahead of the gene, we have a promoter section. Promoter, which is just a strand of DNA that is a attachment point for the RNA polymerase. Right, that's all. And then this right here, this stretch of DNA is the actual gene, right? This is the gene. And the gene, if you remember, and you really remember what we talked about last time, maybe it's got an exon here, exon 1. Maybe here we have an intron. Maybe here is a second exon, exon 2. Maybe here's another intron, and maybe here's a third exon. Right? Because eukaryotic genes have these weird non-coding regions called introns in them. Right, we learned about that last time. Okay. So, but we have one gene. This is one gene that is eventually going to only make one protein. And that gene has a promoter, and the promoter is the attachment point for RNA polymerase. RNA polymerase, which is going to come, and we're going to explain, this is an enzyme, right? It's an enzyme, and it needs to attach here and then move along here in order for transcription to happen. But the RNA polymerase needs help attaching to the promoter region. It needs a couple of things. It might need some proteins that come from outside the cell called transcription factors. Transcription factors are proteins that are separate from the DNA strand, from the promoter DNA strand, from the gene DNA strand. They are proteins that have to come and sort of fit themselves on to the promoter region to make the RNA polymerase able to bind. So that's one thing that eukaryotic genes need in order to activate the making of a protein. Transcription factors, and they can come from hormones from outside the cell that activate them, or they might they might even be self-regulating signals that come from another part of that same cell. So that's one thing that activates transcription. The other thing might be way upstream over here. You might have an enhancer region of the DNA that will fold itself over and attach to the promoter also. We had a couple of different animations that showed you that last time, but if, again, there are special activator proteins that come along and bind to the enhancer region, right? So that promoter needs lots of things sticking to it and changing its shape before this RNA polymerase can come along and successfully bind to this and move along and begin to transcribe this gene, right? So transcription, the first step, transcription, is going to transcribe this into a piece of messenger RNA. Messenger RNA. And that messenger RNA is still going to have, I'll draw it as a rectangle as well, right? Is going to have those exons and introns still in it. It's going to have regions that are exons and introns, exons and introns, because it's just a transcript, right, of that DNA strand. So this is your pre-messenger RNA that has not been edited yet. Once you get that done, you know that in eukaryotic cells you can edit this. You have editing and splicing that happens. And what is it that comes out when you splice a pre-messenger RNA? Anyone in there? Anybody out there? Anybody in here? What comes out? What stays in? Come on, guys. Do the introns get spliced out or do the exons get spliced out?
Go out.
Yes, that's it. Very good. Okay, so when you have your final transcript, it's a little bit shorter because now only exons 1 and 2 and 3 are left, right? And then is there something tacked onto this end? Yes, indeed. There is a cap at the 5 end that helps in the binding of the messenger RNA to the ribosome. And there is a polyadenine tail added to this end, which attracts the digestive enzymes in the cytoplasm and keeps the messenger RNA from being digested. That's basically what it does. Okay, so finally, after translation then takes this in that final step and makes the final protein, a final polypeptide, right? You get one and only, not 2, 1 protein after you fold it and edit it and do all of that, right? Okay, and there's eukaryotic gene expression, where a gene is expressed and you go through the whole thing. All right, so lots of places in eukaryotic gene expression for things to either be stopped or altered or edited or changed, lots of different places for that. And transcription completely different and separate inside the nucleus from translation, which happens way out in the cytoplasm with the ribosomes. So that's eukaryotic gene expression. There. Oops, what happened to my protein? It left. Protein. There. 1 protein. Okay. I don't know why that did that. Maybe I'm going to find a squiggle down here I didn't have before. All right. All right. So now here's prokaryotic. Gene expression. Okay. Prokaryotic gene expression. I'm going to draw another rectangle that represents the DNA again. Right? Once again, we have a promoter region. A region of the DNA where the RNA polymerase, same enzyme, needs to come along and bind in order for things to move forward. Okay, here's the difference. With bacteria, remember we're really talking about bacteria here. Talking about bacteria here. Very, very simple little things. Simple guys. There's not just one gene downstream from the promoter. There's often many. So, for example, there might be a gene here, gene A, gene B, and gene C. 3 genes. And there also are strange binding sites around the promoter called operators, which you won't find in a eukaryotic DNA. There might be an operator site. Okay. So all of this is called an operon. An operon is a cluster of genes whose proteins need to be produced together because often they perform one single job together, right? Proteins are needed all at once. And the Lac Operon that we'll look at is called the Lac Operon because it makes the proteins that digest the sugar lactose. So it's nothing real mysterious. So the Lac Operon that you'll study, that you'll look at a video of later, the Lac Operon organizes the genes for the enzymes, right? One gene, one enzyme. And those enzymes will break down lactose sugar for the bacteria when lactose is present. That's all. That's all. And an operon allows a bacteria to get all of those proteins produced together at the same time without any more than a single switch. Okay, so you notice there are some differences here. There's 3 genes for one promoter, right? In eukaryotes, only 1 gene for each promoter. There's these weird operator binding sites, which weren't there in the eukaryotes. You have these clusters of genes organized together, and the whole thing is called an operon, right? When you have clusters of genes per one operator. You still have a promoter. You still have RNA polymerase that needs to bind to the promoter, so that's similar. But you should also notice is there's no introns here. There's no introns, there's no exons. It's all just genes that get made, right? So when they're transcribed, they just go right to translation. There's no editing of any kind. Also, another difference is instead of enhancers upstream from the promoter, you have something called, I can't ever write this in this. I'm just going to say this is a regulatory gene. So this section of DNA is a gene that constantly makes, through transcription and translation, some sort of regulatory protein. And often this in the one example we're going to see, this is a repressor. A repressor protein is made by this regulatory gene that binds to the operator, right? That can bind to the operator and halts transcription. Right. Okay. So prokaryotic gene expression is kind of all or nothing. It's all or nothing because it all hinges on this guy right here. This guy right here winds up being the all or nothing switch. He's either on or off. Transcription regulated by whether this repressor is on or off, right? Is it here? If it is, there's no transcription, right? If this isn't here, if this guy releases it, then you'll have transcription because there'll be nothing in the way of this RNA polymerase to attach and go head along and do its transcription. make the RNA. And then translation literally happens before transcription is even over. Translation, since you're floating in the cytoplasm anyway, the ribosomes come along and attach to the RNA before it's even finished being transcribed, and you get proteins immediately. And you get 3 of them. In this case, not just 1, because you've got 3 genes there that are making 3 separate proteins. Yes? So about all the regulation that happens here is what's going on with this repressor. Really the only point of regulation in a bacteria is pre-transcription. Because once transcription stops, it just goes all the way through to the proteins, and all that energy is just going to be spent. There's no way to stop it. No stopping. Once it gets started, that's it. Okay, and that's a beginning explanation to this world of prokaryotic gene regulation.